Okay. So we're 2 minutes after. We have a really stacked panel today. So I think we're going to go ahead and get started. My name is Jessica. As I mentioned, I'm a senior program manager for Cure Research at AVAC, and I'm joining you from the DC area today. And we are very excited to have three fantastic speakers who will be joining us. And at first, I'm going to hand it over to Dr. John Frater, who is a professor of immunology at the University of Oxford. He is the PI of the Rio trial. And what he doesn't say in his bio is that he is one of the best translators of HIV cure science, in particular, immune-based approaches, than anybody that I know. So we are very excited to have John talk to us today about the immunology of HIV cure and what's going to be presented today and how we can understand it. So, Dr. Frater, I'm going to hand it over to you.
Thank you. This is amazing. What a great thing to do. I mean, if it wasn't 7 in the morning, it'd be even more amazing. But it's fantastic to be here, and thank you for that introduction. I would just say you very nicely said I was the PI of the Rio trial. The Rio trial is a massive undertaking with lots of people, and definitely the leadership is shared between myself and Sarah Fidler and some other great people in the UK and the US with Michelle Missensweig and Marina Kaski. So, no, by all means, not taking the credit on my own for all of that. I think the aim for today, if I understood properly, was just to run through kind of, Jessica, what you thought I think is interesting at the moment in Cure, and where CROI might be kind of adding kind of value to kind of what we think may be going on. Does that kind of fit what you think we should be talking about?
Absolutely.
Okay, that's great. Well, look, I can certainly give you my perspective on what I think is currently interesting and where we want to sort of focus our attention. Um, so I think that there's probably three kind of main areas from my point of view where I think, in this push for a cure, which has always been this sort of this magical dream out on the horizon. So many people have told us that it's going to be impossible, but I think it's really good. And we heard a lot of really nice talks yesterday about persisting, when things feel up against you. And I think the cure field is a really important one of those, because there's lots of times we're being told we shouldn't, there's we could probably stop looking now, but actually, I think there is such a strong push from community and elsewhere that this still really matters, even in the context of highly effective PrEP and great therapies and kind of the potential for outreach. But I think in the current climate where so much is in jeopardy, I think really trying to kind of push all our boundaries is really important. And I think if we sort of nail down to the kind of the science around kind of how we do that, I don't think there's going to be any sort of amazing moment at CROI this year where we go, oh my gosh, this is really changing how we think about things, and takes us massively forward. I think what we are seeing is lots of small baby steps, and I think sometimes that's as important as the great leap forward. It's building up that evidence that gives you the confidence and the strength to think that that there's movement here. We're moving in the right sort of direction. And I think if I was to break it down into three things, I think the idea of the broadening neutralizing antibody, these mythical beasts that can control HIV as part of your immune system, antibodies that are really good at blocking HIV, where we know that most antibodies that we make aren't that good. And the technology has now moved forward so far that we can identify these antibodies. And we know it's now safe to give them to people with HIV. And so there's a number of trials that have been giving these antibodies to HIV. And one of those trials has been presented. It's a combination of an antibody with another immune agent. But I think more importantly than that, what we're learning about is how these antibodies actually work. And they work differently from a drug. You take a pill or you take a tablet, it does a job, it suppresses HIV or it does something else. And it does that one job and it does it well. What we're starting to learn about these antibodies is that they do more than one job. They do the thing that we think they're going to do, which is to block the virus. And that's great. That's really important. But then after that, something else happens. And it looks like these broadly neutralizing antibodies are kind of talking to the immune system. And they're going, listen, we can actually control the virus better if we work together. And they're helping the immune system, which previously wasn't very good at looking after your HIV, because the HIV was very good at running away from it, from escaping it. And the broadly neutralizing antibodies seem to be allowing the immune system to catch up. And we know that actually the immune system is the best tool we've got. We've been building our immune system for hundreds of thousands of years. So it's an amazing tool. And if we can get it to work as we need it to, then actually it could answer lots of the questions. And it looks like the broadly neutralizing antibodies are allowing it. It's like a facilitator. Broadly neutralizing antibodies are allowing the immune system to do the job that we've always wanted it to do. And there's this is something called the vaccinal effect. It's like the antibodies act like a vaccine. And there's a number of presentations going to be occurring in the next couple of days, just digging into that vaccinal effect, saying, how is it working? Is it working in everybody or just some people? Which bits of the immune system do we need to wake up? Is it the T cells? Is it something called your NK cells, your natural killer cells? And we know that antibodies like have two halves. They're shaped like a Y. And the Y end is the bit that binds to the virus. But the tail, the bottom bit, which we kind of have always ignored a little bit, that encodes all this incredible instructions to do stuff once it's bound. And it's the doing stuff. It looks really exciting. It looks like it's telling, that tail of the antibody is telling the immune system how to behave. So I think as we learn about that, that's going to be really important. And then building on that, just staying with antibodies for a second, we know they're really hard to make. You can't just kind of give some HIV to somebody and go, right, make an amazing antibody and we'll use that as treatment. You have to find the tiny little B cells hiding somewhere that one day might be able to make a broadly neutralizing antibody, but can't do it yet. And this is something called germline targeting. And germline targeting is really exciting because if we can identify early on those cells that one day in the future could make a broadly neutralizing antibody, that can change potentially the way that we can deliver these therapies in the future, which again would be really exciting. So I think that's one thing. I think two other things is one is actually learning more about the reservoir, this pool of cells that contain the sleeping HIV. So while you're taking your tablets, the tablets are working really well on any HIV that wakes up. But we know that behind all of that, there's this pool of cells that contain the DNA of the virus. They don't contain the virus. It's just the DNA of the virus, the code that allows that virus to wake up and cause trouble. And what we're learning about is how that reservoir can potentially be controlled better, and why it's there in the first place. I mean, how come we've got a reservoir? Why hasn't the body got rid of it? And it turns out the reservoir is actually quite good at being resistant to our immune systems, and we're only just finding out about that now. And so learning about how this reservoir is resistant to our immune systems is really important. And there's some really lovely presentations coming out on that. And again, decoding that. If you can work out how the reservoir protects itself, well, then you've got a strategy. Say, well, we can take away that protection, we can knock down those walls of defense, and then actually start to target it. So that's really exciting. And then I think the last thing, which is a little bit more niche, is some of you may be familiar with this whole idea of kick and kill. Which is the idea that you've got this reservoir which is sleeping. You can't target it because it's fast asleep and it just looks like an ordinary cell. So you've got to wake it up. You've got to kick it. And then it shows that it's infected. And then when that cell shows it's infected, you can kill it with something. And we've tried lots of kick and kill things in the past, and they haven't worked really well. It's all been a bit blunt and hasn't been very targeted. But and sort of people said, forget kick and kill, it's not working, let's do something else. But there's been some little bit of sort of chat in the background about, can we do it better? Maybe it's the right thing to do. It's a good concept. It makes sense, doesn't it? You want to kick this thing and then kill it. That kind of makes sense. So maybe we weren't just doing it well enough. And then there's some presentations on how you can make that kick really precise. And rather than just trying to kick everything and hopefully hope some HIV wakes up somewhere, it's saying, well, let's specifically try and kick just those cells where the HIV is, and then that might work better. And then there's some presentations around that. So I think that's probably, Jessica, the three things that I'm excited about: how the BNABs are working and what we can do better, learning more about how this reservoir is resistant to our own immune systems, and then working out if things that we thought in the past that weren't working, like kick and kill, we should give them a second chance and just say, can we do them better? And I think there's some posters, there's some talks, lots of oral abstracts tomorrow and Wednesday on that being presented. And I think, as I say, none of them are going to change the world, but I think they will take us these steps forward each time, and every every marathon is many many steps, isn't it? And I think, and you have to take those steps. That's part of it. You're not just going to jump to the end. So I think, all these steps are going to be really critical and really exciting, for future things. So I think that probably sums up where I am for the moment. Happy to take any questions or pass over to someone else. Jessica, you're in charge.
Oh, I'm never in charge, son, but thank you so much for that fantastic summary. I think what we'll do is if people could put questions in the chat, I think it would be good to go through all of our speakers, hear their exciting, their takes on what's exciting, what to look for at CROI, and then we can have this broader discussion. But I wrote down some pieces to follow up on. So thank you, John. Next is Dr. Katie Barr out of the University of Pennsylvania. She is a professor of infectious disease. She leads the HIV cure research subgroup, working group of the ACTG, and is also one of the most fantastic communicators I know about the cure science. So we're interested to know, what are you thinking about? What are you excited about cure at CROI?
Thank you so much, Jessica. You made it home?
I did. I did. You can't tell from this background, but I am actually in the DC area. Yes.
I think that is a superhuman power that you beat the nor'easter, somehow travel through an insane blizzard. I'm from
Just to get to you
Philadelphia, and my
today,
family
this
is
morning.
texting me pictures of 18 inches of snow right now. So that's not very far from where Jessica
No.
landed in a plane, apparently, and probably slept for about an hour. But anyway, thank you for facilitating this. And I am very pleased to follow John. And I just want to say, John Frater gave one of the introductions to cure talks at the new investigator workshop yesterday, and it was so good. And so I'm very thankful for how amazing that talk was. And I have heard Mr. Dr. John Frater, so many people tell me yesterday how really excellent they thought the talk was and how accessible it was and how motivated and excited they were from the talk. So that is 100% and 110% of what we were hoping for. And so that was awesome.
Thank you. That's lovely.
So I also follow on John's description of the fact that there's going to be a lot of stuff at CROI about BNABs, and not just the fact that they have neutralizing activity and they can suppress virus when they're given in the right context, but the fact that they have these extra potential possibilities to work with the immune system to make people's own immune system a little bit better. And so I'll just highlight the fact that there are going to be a couple studies of post-intervention controllers or people after they were in a part of a BNAB trial, like for example, Ole Sogard's E-Clear study, where they got BNABs at the time that they started ART initiation. There's one individual in that trial who has maintained virus control off any antiretroviral therapy or intervention for years and years now. And I'm gonna try to remember 1 second. They're presenting sort of the detailed mapping of what immune responses are helping to control that virus in that individual. And what they see is that there's both antibodies and T cells that are contributing in really unique way, like the person's own antibodies. And so that'll be, sorry, I think Tuesday during the oral abstract session, that number, abstract 135. And so that'll be a nice, it's one person, but it's a good example of the best case scenario of of early art initiation plus a good BNAB in a person. What's the best we can hope for? So that's it. The thing I want to say about BNABs is that I personally have been working in the field for a while, and I think there's a lot of potential with BNABs. It's not 100% clear how well they're going to do in all the contexts, but I think they could be a really powerful tool for cure. And I think the field's at a little bit of a juncture right now where we had really been developing them in many different ways, and the funding streams have sort of slowed down for BNABs. And so I think we as a field need to think about, there's a ton of priorities in the world right now of HIV, and is the continued development of BNABs, making new BNABs, making discovering new BNAbs and developing these BNAbs. It basically takes specific types of manufacturing to be able to make enough of a BNAb to use in a human clinical trial. And right now, there are not that many organizations willing to fund that. And so I think we as community and researchers, we need to decide if that's a thing we think is valuable and if and how we're going to advocate for that. So that's something that's on my radar of like what I need to think about for the next year. I have like two other buckets of things I was hoping to talk about. One is that in this continued theme of, there's a lot of funding challenges. There's a lot of changes in the past 18 months. And all of the interventional cure research we do, all of the work we do in cure sort of sits within this larger context of how well are people with HIV able to access treatment, able to have antiretroviral therapy, able to have the standard of care that we know is so essential. And obviously that's really threatened around the world in many places. And so even though it's not directly cure, one of the things for someone like me who's not as deeply knowledgeable about delivery of care and each country's individual financial framework and their relationships with et cetera, I'm really looking forward to hearing about the sort of details from experts on this at this meeting. And I think that it's actually incredibly relevant for the cure agenda, because if we can't provide standard of care, life-saving antiretroviral therapy to people with HIV, we can't continue the development of cures very well. And so I think 4 p.m. today, there is a symposium talking about sort of global issues in terms of just delivery of antiretroviral therapy. And I think that's going to be, for me, very important to listen to so I can get an understanding of better what's going on in the world and how do we continue to push cure across the world. And so that's my second comment. And then my third thing is that there are lots of really cool immunotherapy strategies that are going to have multiple different small presentations of different ways of using them. And I think, like John said, it's not going to be one solution, but it's going to be like many ways of targeting specific strategies. So I'm just going to list some of the strategies that are going to happen and that are going to be discussed in a couple different ways. I think maybe yesterday in Sharon Lewin's amazing plenary, she talked about this concept of the molecule TAT, which is a protein that the HIV virus makes, and it makes it to help itself speed up virus replication. And so the thought is that we could use TAT, that same molecule, and deliver it to latent cells that are sort of sleeping under the radar of our immune system and make TAT have those cells show themselves to the immune system. And so there's a lot of groups using TAT. And so Sharon Lewin's group is using TAT. And they've gotten a lot of publicity for these really impressive approaches of delivering TAT via mRNA and lipid nanoparticles. So there's a couple other groups presenting slightly different strategies. And so one of my good friends, Ted Kreider at Penn, is presenting a kind of similar strategy where there's TAT and a lipid nanoparticle. But what they did with the lipid nanoparticle is they put these different molecules on it to target it specific places. And one of the molecules they put on there is called ibalizumab, which is actually an FDA-approved antiretroviral monoclonal. It targets CD4. And so it's a HIV medicine that we have right now, and they're putting that on top of the lipid nanoparticle, and that delivers it specifically to CD4 positive T cells. And so Ted is going to talk about that, I think, Tuesday during the oral abstract session. And that is just one of the ways TAT is being used, specifically delivering it to cells. And it's a nice way to wake up the virus because it's very specific just to cells that have HIV, as opposed to delivering something to all the cells. I'll just mention one more thing. There are a lot of studies looking at this molecule called IL-15. So IL-15 is a cytokine, or it's a molecule that we make in our own bodies, and it's supposed to help make our immune cells work in a little different way, like to go to certain places in the tissues, or to activate and become a little bit more active as they surveil and look around for any cells that may be a little wonky, like maybe they have a virus, maybe there's a cancer, etc. So IL-15 is used as a therapy in a lot of different conditions where we want the immune system to be a little bit more functional. We want CD8 T cells and NK cells to be a little bit more alert and functional. And there's a lot of studies about different ways of using IL-15 that are going to be presented at CROI. So there's a clinical trial looking at BNAbs and N803 or and IL-15. There's in particular there's a study that's going to an abstract that's going to be presented by Jeff Murray from Gilead, looking at a targeted way of delivering IL-15. So again, it's this concept of can we deliver a thing very specifically to some cells and maybe make its effect not touch any of the cells we're not concerned about and protect all of those cells from any side effects and really deliver the thing in a targeted fashion. And so I think those are some of the things that I'm also interested. I'll just say personally, I'm an antibody person, and there's a lot of cool stuff about antibodies. But I think it's a little bit more of what John was talking about how BNAbs and ANAbs together can sort of do more than we had thought of before. And so there's going to be lots of antibody stuff percolating through the meeting. Okay.
And I am just going to ask you to say, what is an ANAB? Because that is not
Oh,
a phrase we always
so
hear.
sorry. I just like said it and didn't even think about it. So BNAbs stand for broadly neutralizing antibodies. And these are these antibodies that have been identified for people with HIV, brought to the lab, maybe engineered a little bit to have a little bit more breadth or more function, and then grown up in the lab as a molecule that we can deliver as a drug. So that's a BNAb. And then John talked about germline targeting, and that is the concept that we could vaccinate or immunize people to make those, to get to those BNAbs. But ANAB stands for autologous neutralizing antibody. And autologous just means your own, like a person's own. And so those are the antibodies that everybody makes against whatever things we have in our body that we are trying to fight. So for a person with HIV, their HIV autologous antibodies are just, I'm sorry, autologous neutralizing antibodies, ANAbs, are just the antibodies they make themselves against their own virus. And they may be broad or they may not be broad, but usually they're more narrow because they just go after some of the viruses that each individual has. And so they're not necessarily going to be able to be taken from one person and given to everybody, but they're specific to the viruses that their immune system has seen in their own body.
Thank you. No, that was great. And I just want to, there's already questions coming in, but I think we're going to go to Allison, Dr. Allison Agu from Johns Hopkins to talk about the pediatric perspective, and then we can bring in everybody to have a more robust discussion. So as I mentioned, Dr. Allison Agu is from Johns Hopkins, where she is both an adult and pediatric infectious disease doctor. She leads the Adolescent Trials Network and is also my partner in crime at the PAVE Collaboratory, leading the Community Engagement Core. So I'm going to hand it over to Allison to tell us her highlights in the pediatric space.
Fantastic. Thank you so much. I did this community breakfast a couple of years ago, and it was a lot of fun to do. It really feels, honestly, an honor to be here to talk with you guys, to be chosen to be here. So thank you, Jessica. And just to clarify, I'm the director of the BTCC, which is a Biomedical Therapeutics Core, but not the whole ATN. I want to make sure I give Sybil and Lisa their kudos. So to talk about cure, I mean, I think it feels like a really full circle moment because I remember being in the room in 2013 when my friend and colleague Debbie Prasad presented on behalf of Hannah Gay and Catherine Luziaga, the Mississippi Baby. And for those of you who don't know the Mississippi Baby, it was a very early treated, an infant baby girl who was realized to be living with HIV with a positive viral load at 30 hours of life and got antiretroviral therapy, which is not what we were doing then, got a full triple regimen and suppressed and then was lost to follow-up. And when she was found back at that point, had been off of medicines for, I think, 14 months and had no rebound. And I remember that being presented in the room and not an intentional ATI, but circumstantial ATI, meaning a antiretroviral treatment interruption. And Debbie presented, and there was all this frenetic energy in the room because before that, there was nothing ever for pediatric cure. It was, there was the Berlin patient and all of that. There was energy, there was hope, but there was also skepticism. Like, what is this? Is it actually a thing? And so and then, about a few months later, when she rebounded after 27 months, there was still hope because we could at least go 27 months off of ATI. And so since then, there's been so much progress in pediatric, cure with everything from the Impact International Maternal Pediatric AIDS Clinical Trials Group, having a whole cure committee that's actually led by Debbie, the landmark P1115 trial, which is done in multiple countries, including many in the global South, where the goal is to try to replicate the Mississippi baby. She's never come forward, but I would love to meet her at some point to thank her for what she's done for the field of pediatric cure. But P1115, early treated treatment, has reported, I think 2024, 6 kids had reached the profile of where they could stop that antiretroviral treatment and see what happened. And 4 of those 6 kids in 2024 had not rebounded. There's going to be some updates from PLM15 at this this conference today, or so looking forward to seeing what's new from PLM15. I think for pediatric cure, it's very early treatment. And so Baby Cure in KwaZulu-Natal, led by Phil Golder and his group, very early ART in infants and have enrolled, I think, about 140 infants today that are living with HIV. And previously they reported on 5 kids that had reached this ATI and not rebounded, and they're going to report some more. I'm hoping it's embargoed, so I can't say. In fact, I don't know. So go to that session. I think it's Tuesday by Gabrielle Cremot. To see what the updates are on that, and in terms of early treatment, it was something that we could do now, right? So I know in many of us in our clinics, we are seeing young babies where there is high risk potential transmission and starting early treatment and trying to replicate that finding. And it pushes for us as clinicians to, one, diagnose early and to treat early and to then say to our young people who are taking medicines and maybe getting tired, but early treatment we know leads to smaller reservoir size, et cetera, and maybe actually put you in the queue for more likely for cure. So it's exciting to see these updates at at this conference today. Catherine and John talked about BNABs. And again, a lot of BNABs in adults, but there's a lot happening with BNABs for kids. The Tutelo has reported on BNABs for kids, I think last year, Corey. There are going to be updates, which is BNAB giving early treatment, already treated children, BNABs to see what happens again with treatment interruption to see if they'll rebound. So Ajibola is presenting today on an update, I think tomorrow, actually, in the same session with Gabby on updates from Tutelo. So really excited about that. And using combinations of BNABs in children. So a lot of times children are the delayed afterthought, and to see what's happening in adults also happen almost parallel for kids is really important to move the field forward for kids as well. There's going to be update on more BNABs 2008 from Impact, that's Tutelo, and other BNAB studies in kids to see, again, updates in how they're doing, including how they clear BNABs that their mothers may have gotten. So very interesting to see. There's some therapeutic vaccine data by Tang and Debbie Prasad's group looking at impacts of therapeutic vaccines on reservoir. I think those posters, it's a whole poster session in pediatrics on therapeutic vaccines and BNABs this afternoon. I think it's P2 and P3 this afternoon. So check that out. I mean, that never happened before. You'd have one scattered here or there. To have a whole session where line after line we're talking about BNABs in kids and therapeutic vaccines and others are great. I do want to highlight that as we think about cure in pediatrics, it's been really important to align with the lifetime survivors and the dandelions who themselves have a lot of not just thoughts about treatment and cure and what's needed, but they also hold the key in their reservoirs and their long-term treatment in terms of how their reservoirs may act and how they may respond to cure. So it's really been wonderful to work with them to have their input and insights on what we do. I would be remiss if I didn't say in all that's happened since 2013 in Mississippi Baby to not highlight that there's actually a pediatric AIDS virus, an actual research collaboratory dedicated to pediatrics, which is important as we move the field forward. And then last but not least, not at this conference, but a shameless plug about Cure Research. Jessica, myself, and our members of our PAVE CAB actually have put out and now published in Journal of Pediatrics HIV cure research, the ethical and real world practical considerations for pediatric and adolescent populations, just really highlighting the life course perspective and how we need to think about the different aspects of what's happening, whether the strategies that we're thinking about, but also how they may affect different perinatally acquired and lifetime survivors from the babies all the way up in terms of cure. So I want to highlight that, Jessica. Hope you could drop that in the chat for people to look at that. But a lot happening here. And as we look at that adult studies, also thinking, and then what would be next for kids and how what ethically, practically can happen. So really excited to be part of the conversation and to go from there. Back to you, Jessica.
Absolutely wonderful. And thank you for the reminder that when we talk about pediatrics, it's really about perinatally acquired because it's the aging, it is the life course. So it's a really important thing you put it there. So I'm going to dive right in with the questions because there have been a number of really exciting things that have come up. And I do want to remind folks, we talked a lot about the immune system and what kind of the exciting factors that can happen there, but there's this other bucket of work which John highlighted and Katie highlighted a little bit, which is that things that are targeting the virus. And so when we really think about curative approaches, it's things that are working to enhance or help the body, the person, and things that are going directly for the virus, oftentimes to either wake it up, keep it asleep, or cut it out. And those two things work together many times in a combination approach. But as you can all throughout CROI, you'll kind of see the one step, and then a couple of times you'll see those two sides coming together in a combination. So with that, I'm just going to start. There was a really wonderful question actually to you, John, with somebody who's living with HIV for 40 years and undetectable for 28 of them. This is from Gus Carnes. I get a bit disheartened by the fact that cure studies always seem to find the newly acquired individuals who start ART straight away. Are there any good news for long-term survivors with single-figured naders? And I would also say that while this for adults, I'm also going to ask you, Allison, if you can comment from the pediatric life course as well, from the dandelion perspective. So maybe John, you first, and then we can move to Allison.
Yeah, I mean, thank you for that question. It is so important, and I think we're all very conscious of of that kind of apparent sort of, it's like kind of refined nature of some of these clinical trials that kind of you look at these data and they go, well, that's not me, and I think that's really important, and it's something as a field we are 100% aware that we need to address because we work very closely with the community in everything that we do. I think that where you're coming from on that, I think the difficulty when you're trying to do something where there's a lot of pushback potentially from funders and advisors and government saying, we could be spending our money doing other things. You start where you can get you're most likely to get what you might consider a good result. And there was these data that came out from Assay Zerion in Paris saying that if you find someone who's been treated early and you stop their therapy or the therapy is stopped for whatever reason, the chance of the virus coming back is significantly less than someone who started their treatment later in life. And so when the cure field, I think, started pushing to say, look, we can make a difference, the focus was on people where we thought it would be most obvious to show that. And there has been, as a result, a bias, in finding people who were treated early and then saying, can we see a difference there? I think we're at that stage now. Well, I know we're at that stage now because we've just opened a trial in the UK, which is for everybody. The Rio trial that you might have heard of, we particularly said we want people who've been treated early because we think if the antibodies are going to make a difference, and it was an antibody trial, that's where we might see a difference, and that'll make us, then we can do the next step. So we've just opened a trial in the UK. It's called ABVAX. It's another antibody trial, but it's combining antibodies with a T-cell vaccine. So what Katie was saying before about, everything coming together, getting the immune system to talk to each other. So you've got the antibodies on one hand and you've got the T cells working together. And that's a trial which is for everybody. It doesn't matter when you started your treatment. We do want people at the moment where their immune systems show some degree of protection. So reasonable CD4 counts, we've called it 350. So that if we're stopping treatment, we'd be nervous that it dropped too far. And I think we need to see more evidence that what we're doing is safe before we kind of we in my mind at least, open these studies up to people with very low CD4 counts at the time and ask them to do a treatment interruption. So I don't think we have the evidence to know that that's safe. And I think the whole area that we're working in, almost all the people we work with, all these amazing participants who come to our studies, they're all really well. And they're doing brilliantly on their tablets. Obviously, there are often, lots of sort of problems around having to do that. But generally speaking, when you stand back, they're doing really well. So the bar for us is really high in not doing any harm, which might be different to some other studies and other conditions. When let's say if you're doing a cancer trial and people are sick, you can take more risks there potentially. But for people with HIV who are doing so brilliantly, the danger of causing harm is so much higher. So we have to be so much more careful. Saying that, definitely moving into people now who've received treatment later in life. And also, just to add to that, there's been this bias, and I think it's very much, I think, in some of the European studies where the people who've been coming forward for trials have been men rather than women. The Rio trial, without any of our, trying our hardest, was all men, which was crazy. We were desperate to get time to ask or for women to take part in that. And I think there are life issues as well. I mean, we're trying to get as many women as we can in. And the idea of coming to regular appointments, it just seems to be a little bit harder, and trying to facilitate things for women to take part in trials is something we need to think much better about. There's a lovely talk yesterday about implementation science. And actually, we don't think about that well enough. How do you make it feasible for people to actually turn up and take part in these studies as well, to broaden them out? So I think the last is there any good news for kind of, and I think there is good news, because I think we're now moving to a stage where we feel like what we're doing is safe. There's still a lot of discussion around treatment interruption, and I think we all feel a bit nervous when we do that, allowing a virus to come back, because it's in our DNA. Not, we're here to prevent virus coming back. So when you do a study where you're allowing that to happen, I think we all feel a little bit anxious that we're doing it safely. So I think there's always someone looking over your shoulder going, is this the right thing to do? And I think in that context, I think we just need to kind of keep going slowly and steadily, rather than sort of charging forward, and hopefully not making any mistakes. So I think there is good news. I think as we move forward, more and more people have the opportunity to take part in these studies, which I think will be really exciting. And then we'll see how well these different interventions may work. And hopefully, as time passes, Kate is saying, more things may come along where we can trial them as well. So yeah, so I think that we are still moving forward. Hopefully there will still be an argument this is a priority. And that's a different discussion to have. And the community may feel that it's becoming less of a priority. It'd be really interesting to know what people think about that. I've certainly had conversations with people who say, look, I'm happy with mine, one tablet once a day. I'm not interested in cure. And I have other people who are passionate for a cure. So, I don't think it's a one size fits all thing either. So I rambled a bit off topic, but I think I hopefully answered some of it.
No, thank you so much, John. It's an interesting piece. I think this idea of how do we target long-term and lifetime survivors. And maybe Allison, you could share a little bit of a perspective from the lifetime survivors, if there's any trials or research that's, specifically targeting that group, and what we could do to enhance that.
Yeah. So I think one of the things that's really important about this kind of research, and I think the questions about, how early you start and when do you start, what's different about lifetime survivors is that we know exactly when they acquired the virus. And so then there are cohorts like the Pediatric HIV/AIDS cohort study, and actually our own cohort at Hopkins, shameless plug, JHG cohort, where young people who have been treated for really early, as well as those who were treated, a little later in life and looking at their reservoirs. And so there's a lot of research happening, whether it's just looking at their reservoir size, looking at their immune responses, and I think they're different. And putting a plug as to why we need to actually look at this group differently, because how they may respond to certain treatments and nabs and wake and shock and kill and all the things that I'm not as well versed in may be different. So absolutely critical. And there are, again, cohort studies, but also in vitro studies. And then those groups also advocating for and should be intentionally included in trial, because many of them have been suppressed for years, have robust CD4 counts, and should be no reason why they wouldn't be able to be in some of the treatment studies or the cure studies as they go forward. So thanks for raising that. I think really important.
Yeah, really, really critical discussion. And I think this is really where community can play a role and advocates can play a role, especially as the landscape for funding is shifting and what the priorities really are. So I thank you so much, Gus. Katie, there were a couple of clarification questions, and I'd love to have you maybe explain IL-15 interacts with other cytokines and also explaining vaccinal effect and what that means. And then we can go into the next question. But yeah, if you could explain those two things for us, I think that would be really helpful.
Okay. Yeah, I think I have a bit of a cloudy head and I'm throwing a lot of terms out there that are not explained without even realizing it. So I'm going to try to do a better job. Can I just pile on what Allison and
Yes.
John saying? I'm not a pediatrician, but I personally feel one of the biggest opportunities for BNABs is in the pregnancy, infancy, and adolescent space. And I think that there's so much potential for these antibodies to be used in these really challenging times to dose antiretroviral therapy, to allow women or pregnant people to have successful pregnancies, to breastfeed successfully, and then to deal with adolescence, which is, in the best cases, a really difficult time. And when you add all of the other concerns about living with HIV on adolescents, if we could make that a little simpler, a little easier, a little less side effects, I think that would be amazing. So I thank Allison and John for both working for BNABs and pediatrics across the line. I think that's such an exciting place. I have great hope for that field. I think all of the research and clinical trials in pediatrics is like 30 times harder than anything that's done in adults, and it's already hard enough in adults. So keep pushing because I think there's so much promise there. So thank you, Allison, for working in that field. It's so vital. Okay, IL-15. IL-15 stands for interleukin 15. And I got to tell you, John is a cellular immunologist, and he understands all of these letters and numbers that get put together in random ways to describe the literally thousands and thousands of components of the human immune system in terms of cytokines and cells and types of whatever. There's literally an infinite number of components of our human immune system, which is why it does such an amazing job and why there's so many ways it can slightly go wrong or slightly do different things. And it's incredibly challenging to disentangle what's going on. So, IL-15 is one of the hundreds of interleukins, ILs, that we have. And specifically, the reason why it has a lot of prominence right now is because we've begun to learn about this molecule in its role in cancer therapy and in autoimmune diseases, because it is believed to be relatively safe and sort of activating for our immune system, as well as telling our cells to go to the tissues. So it sort of makes these immune cells wake up, the T cells, and it sort of tells them go to an area which we think is important for where HIV resides, where HIV lives. And so IL-15 absolutely interacts with all the other millions of components of the immune system. And so that's been part of the process of the past 10 years or so, is figuring out in people with HIV, what does IL-15 do? We think it should tell cells to wake up, tell them to go to the place where HIV lives, and start doing their job better of recognizing cells with a little bit of HIV in them. But of course, it's way more complex than that, and you have to think about when you're using it and what else is going on in the person's life, and is the virus suppressed by antiretroviral therapy or not? So that's been the process of the past, the trials with IL-15, is teasing out how it works in this super complex immune system and figuring out, does it have a role, as Jessica says, to either make the virus wake up, make it go to sleep, or to cut it out, right? I feel like we could have a good dance. And I am an incredibly clumsy person who should not be making that dance, but I do feel like we could have a wake up, sleep, cut dance, and it would be very helpful. Yes, Jessica can drive that project. But so that's what IL-15 does. It is specifically supposed to wake up cells, make them go to the tissues where HIV lives, and then hopefully, make the virus wake up as well. Will it do that? Will it do that? I don't know. In some cases, it seems to be promising. And again, the thing that they're talking about in this abstract today is if you specifically take that IL-15 and you target it to specific cells, can we make the effect of that chemical or that compound a little bit better without any side effect, with fewer side effects? Okay. Was there a second question, Jessica?
So I did want to ask about vaccinal effect and
Oh,
just what does that mean? Because we hear that a lot.
yeah.
And we will hear
So
it a lot over CROI.
So when you get a vaccine, when you go to get your flu shot or you get your measles shot, what we want is to give a little bit of, we want to show the immune system a bad virus or pathogen, and we want to make a good response against it. And almost all of our vaccines, when you see that, if you give a flu vaccine, it's a little piece of the flu, and we're asking our body to make cells and antibodies against that thing in the vaccine, so that if you ever see it again, your immune system is ready to go with antibodies and or cells that recognize that, can see that flu, and can immediately attack. So the vaccinal effect is asking whether BNAbs can train the immune system in the same way. So the thought is that BNAbs, because remember, John says it has that Y, and so this part binds to the virus, and this part down here interacts with the immune system. And so if you get a bunch of BNAbs binding to virus, and then it shows that virus to the immune system, kind of like a vaccine, and it trains the immune system, the cells and the antibodies, to see that virus better and hopefully make responses so that when it sees it again, it'll do a better job of controlling it. And that's the vaccinal effect that John was talking about. These BNAb studies are asking about, in addition to the first effects of the antibody, does it train the immune system like a vaccine to see the virus in new and better ways and be more effective in controlling the immune system later on?
No, thank you. I think that's so helpful. Um, it's this is all complicated science when it's not 7 a.m. And so I really appreciate how all of you guys are just doing such a fantastic job breaking it down, and the chat agrees with me. There are other questions, but I do want to give an opportunity if somebody would like to ask a question, to our panelists directly, please raise your hand. I know I don't always love to hear my voice constantly, so please raise your hand, join the conversation. We'd love to have you with us. Oh, I apologize. I'm on a call, but thank you. While we wait for somebody to join us, we did have a really fantastic conversation about somebody was asking about the difference in gender and sex differences, because we didn't really talk about that. And so this is really open to any of our panelists to talk about what are some of the sex differences, and should we be looking for any particular posters or oral presentations that might highlight some of these sex differences and approaches to cure. And now my son is gone, so we can all, anybody can feel free to answer.
And I think the hand up is just popped up on my screen too. So.
Good afternoon. I am so sorry I can't raise my hand. I can't see the button for raising the hand. I'm Zandile Masangane from the Kingdom of Eswatini, also known as Swaziland. Thank you so much for the so important information. I know that in our inpatient in Eswatini, we are seeing more HIV positive clients who are on ART now, developing the non-communicable diseases in the likes of diabetes mellitus. We are seeing more diabetes mellitus type 2. I don't know whether it's the influence of the dolutegravir, because most of them were transitioned to dolutegravir, seeing that it helps in raising the number of CD4 count. So I don't know whether it's the side effect for that drug, and what can we do as a country, because we are seeing more diabetes, especially diabetes type 2, diabetes mellitus type 2. Thank you so much.
Sorry. I'm happy to, I mean, I think we definitely absolutely or know that HIV itself is inflammatory and immune activating and may have increased risk of non-communicable diseases, like diabetes, hypertension, strokes, and on and on and on. And so, some of this, could this be people who've been in long-term treatment with metabolic impacts of the HIV? Sure. And the therapies. Could this be weight gain related stuff? I think it's multifactorial. I think we're also just seeing higher rates of diabetes, hypertension in the general population as well. And so now you have the intersection of the HIV therapies as well as just what's happening at baseline. So you're absolutely right. We're seeing it everywhere. And I think it's another reason why we also need to be thinking about one, not just the toxicity of our therapies, but how we can actually get to people who have healthy foods and all the things that actually improve weight gain and improve that, but also cure is a way to hopefully avert future or development of other things. So I think it's part of the conversation about that. So thanks for raising that.
Wonderful. We don't have too much time. So Simon, can you please join us?
Hey, thanks. Fantastic talks. I put a quick comment in the chat. I really just jumped out a bit, Katie, when you suggested that a huge area of research should be defunded. It didn't come across really well, particularly how much, the most effective intervention so far to be able to keep some people off treatment has been BNABs. And we know that they're expensive and we know that there's issues that they're expensive at the moment, but if they're produced in bulk, the cost would go right down. But it was more taking out a whole side of research that is really vibrant. It must make any researchers hearing your comment there that their field should be defunded, I don't think is really helpful. And as a community person, hearing researchers argue for funding against each other really doesn't really help. And Sharon Loon's talk last night went through more than 10 years worth of interventions to try and agitate the reservoir and to wake it up. And we're not suggesting that we don't do that continue that research, even though none of those interventions have been any use at all to any of the people in the studies. So really just a tempering of that comment did jump out a bit, and I thought that was a bit unhelpful. But thanks for all the other talks. It's they're really impressive and a really good way to understand CROI. Thanks.
Thank you, Simon. Can I guess I attempt to clarify. I in no way mean to suggest that we should, or I mean, what I'm hoping to suggest is that I think there's a ton of promise in BNABs. And what I am concerned about is that for a long time, the US government, the Vaccine Research Center, a lot of programs funded through the US government have paid for the development and discovery of new BNABs and the production of BNABs so that this work can continue. And right now, that funding for continued development is very uncertain. And so what I'm suggesting is that we as a research community, we as a community of advocates and cure research supporters need to continue to make our voices clear that this is a promising area. It's not that the research would be shut down, it's just any of these research projects require somebody to make the intervention, to make the BNABs. And John, you could potentially talk about where you've gotten your antibodies, but a lot of the antibodies for the Rio study, for the eClear study, for all of these big studies that have shown really promising effects have essentially been funded through the US government because they are contracts to make the large amounts of antibodies like that Michelle Newson-Swag has made in order to fund a lot of these studies. Those were supported by the US government, supported by grant funding. And, we are in a place where there are some promising new antibodies, and some of them are being managed by drug companies, which is great because we need pharma to be involved. But there's some others that are in academic research labs right now, and they're searching for a path to make those available to the world. And so my hope is that we actually have the next versions of the best BNABs out there, but they don't yet have that level of financial support that they need to bring them to the clinics. And that's what I'm hoping we can advocate for. I don't think anything is going to be shut down. I'm not trying to pit anyone against each other. What I think we need is to say that we believe this is promising and we want continued support to make the next versions, because the BNABs we have are good, but we need the next generation. And some of them have already been described, and I think they're out there, and we just need to keep pushing that support. That's all.
Yeah, and maybe I'll add, just because this is a really important piece, that there were a couple of questions that came in about diversity, sex and gender, but also clade differences. A lot of the work has been done in the global north, particularly in Europe and the United States. But more and more that is shifting into sub-Saharan Africa, into Asia, where we do have different clades and where we do need to think about the sex differences in different ways. And there are research that will be presented at CROI that isn't necessarily going to try and highlight the sex difference or the clade difference, but is being done in a different region. So we'll do so. I know Allison had mentioned the work of Philip Goulder and that lab. The previous pieces that were reported on were all boys, all males that were in that cohort. Who know, and I think if you go to that session, perhaps that might be different. And that's something to think through, those gender aspects. The work that's being done there is also in Clade C. So there's just a breadth of research that may not highlight diversity at its core, in the title, but at its essence is really talking about the ways this field is diversifying and what we can do as advocates to talk about and continue to push that, because we will need a combination wherever we are, a little bit focused on the body and a little bit focused on the virus, and how do we bring those two together all over the globe. And so there's going to be a lot of that research here, those steps, and we're going to be the ones to help put it together. Allison, I see your hand, and you are also going to start with our last comment wrap up. So please highlight, and then I'm going to pass it on to each of the presenters.
No, definitely. So my comment was going to be picking up on a question or a comment that Bridget Pico put in the chat about women, including women in studies. And I think this goes to not just women, but diverse populations in trials, and how we assure that our trials include diverse populations, and making sure that we ask community to weigh in on what's needed to participate. Because I don't know that it's hardly it's hard to reach. I think it's hardly reached in a way that they want to be reached and reached and engaged. So I think that's really important. I wanted to lift that up because we're not going to be able to identify these differences if we cannot get people to participate, because we are making it inconvenient or what have you for them to go to participate. So engaging people, hearing what they need to effectively participate, and designing trials in a way that help to support them.
Great. John, and then Katie, love your final thoughts.
Yeah, this has been an amazing conversation, and it's good to trash these things out. I was just thinking about kind of what Katie was saying and that comment from Simon. I think we are in a difficult position. I was trying to think if there's an analogy to the history of antiretroviral therapy, where are we in BNABs? I kind of feel like we're probably at that 2-drug stage. We're giving DDI and DDC, and we know it's kind of works a bit, but it's just not good enough. And the problem is getting past the PI stage and into the integrase inhibitor stage where we are now. We're a long way off that. And the studies that are being presented, they're generally 2 antibodies that were made by the same lab, and now being bought by a company. And it's just been, it's gone into the too difficult box. Where do we get the next broadly neutralizing antibodies from? And it's a word broad. What do we mean by that? They're not that broad. Yeah, we need extremely broadly neutralizing antibodies. There needs to be a new, label for this, because they don't cover they don't cover all subtypes. They don't cover all viruses. 1 in 3 people are probably resistant to an antibody before they've even had a chance to see it. So we are in these early stages. So I think we need to make the case, as Katie was saying, that to do something really difficult, which is going to be expensive and problematic, is worth investing in, and, in time when budgets are difficult. So yeah, I think it's just a difficult time, but it's about pushing against the tide of it, saying, we need to kind of invest in these because there is hope. And the data that's coming out in CROI suggests these can work. If 2 drugs work, 3 will work better, sort of argument that we had 20 years ago. That's kind of where we are now.
Thank you, John. Katie, over to you.
Okay, thank you so much. It's really wonderful to be here with you. I just want to, yes, I think we need to continue to do a much better job in diversifying our clinical trials. I'll just say from the ACTG, which is one of the major US clinical trials group, we have a couple studies currently in Southern Africa that are either majority or almost exclusively women, because when we work in Southern African clinical trials units, we have more ability to connect from the site to the participants to make that to enrolling women reasonable. And so that has been quite successful. And we're trying really hard in our US sites to do a better job, because I agree, these participants, it's a failure of the clinical trial sites, not and the study design, not the participants who very reasonably say, you cannot ask me to do that. That's insane. And so we need to make asks of people that are more reasonable, and we're working really hard to do that. So we have a bunch of initiatives to try to work with sites and communities and like the community groups that work largely within different groups to figure out, how do we make trials more reasonable for women? How do we support them through it? And hopefully we can get from our, in the US, we tend to see like 10 to 20% women enroll in our cure studies. If we, in the recent past, when we've been trying really hard, and we have a couple new approaches to try to do much better than that. Yes, so it is really a failure of our trials and not a failure of any individuals or communities who are actually quite engaged, but can only do so much with their complicated lives. And I've been reading the chat, and the chat comments are very insightful, very thoughtful about the burdens that women face and how we need to meet them part way, because they are definitely willing to participate if we make a slightly reasonable ask of them. And so that's a great thing. And I appreciate, the last thing I want to say is, I recognize many people were not able to come to CROI for visa reasons, for funding reasons, for all of the difficulties that are in the world right now. And it's so wonderful that people are still engaging, so committed to the research and the work, and the continued sort of involvement. And so I appreciate that effort. And I hope the virtual platform works well enough that everyone can still feel involved. And I think from the CROI organizing people perspective, it would be great to let them know what worked and what didn't this year, because as they figure out moving forward whether things will be in the US or outside of the US, this is probably going to be an issue for the next few years. And so we really want to continue to involve as many people from around the world as possible. And if there's any thoughts or comments about how that can be done better, I know that there's a strong desire to figure out how to make it as reasonable as possible to continue this dialogue, even with all of the restrictions.
Yeah. Well, thank you guys all so much. Thank you to our amazing panelists. I learned so much every time I talk to all of you, and to have all three of you in one Zoom room has just been such a delight for me. It's just been fantastic. And I know others on the chat have felt the same. I want to thank just the co-organizing sponsors here, EATG, Fiera Cruz, TAG, and AVAC for putting these breakfasts on together, the Community Liaison Subcommittee of CROI. And I hope that everyone on the call can join us tomorrow for the prevention talk. It's going to be equally as riveting and exciting, and we'll preview all of the amazing prevention science that's going on at CROI. So thank you all so much for your time and for joining us today. And we really look forward to staying engaged. Thank you.
Thanks, Jessica.
Thank you.
Thanks all.